Hi, everyone. Welcome to the next episode of Editors in Conversation, the mBio edition. This is a show about the tiny organisms shaping our world and the scientists that are uncovering their stories. I'm your host, Marvin Whiteley. I'm the editor-in-chief of mBio. And today we're actually looking up. We're actually literally going to be looking up, and we're going to be exploring the microbial life that's in and around us and above our heads in the atmosphere. And I chose this paper. I was really intrigued by it. And one of the main reasons is when I have conversations with scientists and non-scientists about the microbiome, almost the very first thing that people think about are themselves, and they think about the microbiome in their guts. Maybe they actually think about the soil and the oceans a little bit, but it actually turns out that the atmosphere around us and above our heads has a dynamic community of bacteria and fungi, and then the atmosphere is actually moving these microbes around. And so today we're going to discuss a new mBio study that was published January 2026, looking at airborne microbial communities in a subalpine forest and a grassland in Colorado. And I'm delighted today to have two of the study's authors here. We have Carolyn Cornell and Jane Stewart, and it's nice to have you. And welcome to the podcast.
Great, thank you.
Thank you.
So for us to get started, I'd really like to start just you guys briefly introduce yourself, kind of tell us, where you are and what you've been doing, and maybe we'll start with Carolyn.
Okay, yes, I'm a postdoc fellow with Jane at Colorado State University. I'm here on a fellowship too, where I work a bit with the U.S. Forest Service. So looking at pine plantations, but also looking at the soils, the microbes on the plant, as well as in the atmosphere. And my background has mainly been bacteria, but luckily Jane's helping me focus more on fungal communities as well.
Okay, cool. Jane?
So I'm a plant pathologist. I study primarily fungal systems, and I study pathogens in trees, and that can be natural forests, urban trees, or in crop settings. And one thing that got me sort of started in the microbiome was looking at how fungal spores move around in the sense of what's the risk for disease development. And so that's kind of what we do a lot of different things in the lab, but that's one thing that we've started to focus on.
Okay. Yeah, when I read the paper, again, I do a lot of microbiome work, but obviously not in this field. And I saw this word aerobiome, right, that you guys were using. So I don't know, Jane, can you tell us what is the aerobiome?
So it's basically the collective community that's in the air, right? And that can be bacteria, it can be fungi, it can be protists, it can be anything that we see in the air that's microbial and biotic. And we try to pull out the plants, right? There is pollen in the air, but really when we're thinking about the aerobiome, we try to focus on the microbes that are in the air.
Okay. And so, when I first started thinking about this, I was thinking about, there's definitely microbes there, but are they metabolically active? And what are they doing? Are they just kind of sitting there? Or are they growing? Are they dividing? So what's going on there, Carolyn?
Yeah, that's always a big question in this area of research. It's definitely ongoing. We obviously focused more on who's there, not who's active, but that's a big portion of it. A big way people do that is looking at it. We know that like ice nucleating particles, and they work as condensation nuclei, so they can kind of help form precipitation in clouds. So we do know that they're doing things in the atmosphere, but they definitely are not there as long as maybe they are in other communities, like in the soils or in the ocean. So it's both, most likely, that's kind of happening.
And so the aerobiome can impact the weather, is that right?
Yeah, it definitely can. That's a big portion of where we're looking at it from an activity standpoint.
Okay. And so I guess that's one reason why, as microbiologists, that we should care, but why else should we care about the aerobiome?
It's really associated with one health, right? It's associated to our own health. So we know that allergens can move across the oceans and start off in one location and move into other locations, right? So, human health, plant health, the weather, the aerobiome affects many different aspects of our lives, for sure.
And when we think about, I was thinking about, when I study the microbiome, maybe the oral cavity, you take a little saliva, or you just, you swab someone's mouth, or obviously with the gut's pretty easy to get a sample also. But for you, how hard is it to get? How do you sample, right? How do you sample the air? And
so we basically put filters on fans that suck air at a really high rate. up pretty high. So we were able to use towers that were the highest was about 80 feet into the air. And you set up one of the filters in the machines where it's sucking the air onto the filter, and that traps the microbes. And then we take those filters and then we able to extract the DNA and then amplify basically what the microbes are. But you have to get into the air, and then you have to sample the air for a long time because the density of microbes isn't that great. And so when we think about the downstream effects, we have to usually sample for about 12 hours at a time to get enough material to be able to extract enough DNA to be able to sequence what that is, what microbes are there.
And so in your study, like you said, you went up like 24 meters, right? I think that was the height, the tallest that you went. Have people gone higher than that and actually looked at what's higher than that?
They've actually done it from airplanes. And so they're able to collect high up in the troposphere and see what's there. And they basically see a lot of different microbes, even up that high. It's pretty diverse community up there.
Okay. And so, but in your study, you sampled at multiple heights. I think what was it, 2, and then what was the
2 meters, 9 meters for across both sites, and then the tower was higher at our forest site, so we were able to get a sample pretty much above like the height of the trees.
Yeah, I was wondering, just not in the field. I mean, why do you choose those heights? I mean, is this been empirically looked at, Carolyn, or is it? I mean, what is the reason for this?
I mean, partly availability of the towers and what we're able to make work, but then you also consider kind of the vegetation at these sites and sampling kind of within them or above them. And really with the forest, you can kind of be under the trees, within the trees, and above the trees, where the grassland vegetation isn't quite as high, but it's kind of where you kind of get mixing, and we can see differences based on what height we are in these different kind of ecosystems.
Mm-hmm. And I was wondering also, does the number of microbes change as you sort of start at the ground and sort of begin to move up into heights? Does the numbers change? Do the numbers of microbes change?
It definitely does. Isn't something we specifically looked at, but you can think of from a mixing standpoint or from the source availability. You can get more coming off the soil the closer you are, and then it's going to become more selective pretty much as you go up. The pressures that exist are going to usually become more intense as you go higher into the atmosphere.
Yeah. Okay. And so, yeah, when I was looking at the paper, I thought it was really interesting, sort of the questions that were there, but I think it might be useful, I guess, for the listeners what was the motivation, right? I mean, this looked like a lot of work. It just, when I was looking at it, I was thinking, wow, this is a lot of work. And, it's hard work too, just doing the sampling. So what was the motivation here, and what was the gap that you guys were trying to solve in the literature?
I guess the motivation was to try and determine if source, right? So if we think of ecosystem as our source, we've got the grassland and the forest. Do we see different communities coming off of these two different sources? And then one thing that was really unique about our study is we were able to look at 2 sites that are only, I think it's 60 miles apart. And so we were actually able to look at, are there things traveling from one site, from the forest to the grassland, or from the grassland to the forest, knowing what was our source community, and then we could sort of backtrace what was present in both. And I think that was something, there have been studies that are looking at source, like different ecosystems, urban or farmland, but to have the 2 together, I think was a pretty unique portion of the study or aspect of the study.
Yeah, I thought, the spatio-temporal aspect was pretty cool, right? That you could actually look. And so these were 2 sites 60 miles apart. How big were the sites that you were looking at?
Well, it's the tower, the tower, how big are the towers? They're not very big, but the sites are huge. I mean, this was a forest ecosystem and then a grassland site. So they're pretty big. I don't know the range that we would call them, but they were basically a forest ecosystem and a grassland ecosystem. Like these towers are just right in the middle of them. Yeah.
And, these communities that you see, I guess, my first instinct when I thought about it was, what you would see at maybe, 24 meters is really impacted and really reflects what's below it. Is that true, or is that not true?
Yeah, Carolyn, you want to hit that?
Yeah, a little bit of both. Definitely more so in, let's say, like the forest community, because you have your vegetation that's a lot higher, and a lot of the microbes can be lifted off this vegetation. that it would be very reflective of the site. Compared to the grassland, you get a lot more input from other sources because the sources that it's coming from tend to be much lower, closer to the ground. So we saw that ecosystem type made a huge difference of what we were seeing at kind of each height. And we did kind of look at what showed up between both of our sites. Most of the things that we saw higher up in our sites were what were shared. So that's probably whether it's transferring from like the grassland, from the forest, to the grassland, or just from other air masses moving across both sites. At the higher up, you definitely get a lot more similarity and sharing between these communities.
And sort of this temporal aspect, I mean, and I'm sure you guys probably did this by just reducing your data sets down. If you just would have sampled once, what would you have gotten? If you wouldn't have done this over time, what would you have missed in the study?
I think we would have missed basically organisms that perform better in cooler versus hotter environments, right? So we were able to see that July was pretty different compared to the other June and October. And it's probably because hotter temperatures, hotter soils. And so I think we would have missed different communities. They just wouldn't have been present.
Okay.
Different taxa.
Okay. So different times of the year, the air that we breathe has distinct microbial communities, right? Is that right?
Yeah.
Okay. Yeah, definitely.
And does the ratio of sort of, I know you found fungi and you found bacteria, and we'll get to this in a minute. You found some cool things that I think were different between those. But seasonally, do you see different proportions of that? Like, are there more bacteria, in the fall or in the summer than fungi, or is it about the same kind of, throughout the year?
I think it definitely can differ of what, favors fungal sporulation compared to bacterial communities. Like a wetter community or more humidity in these sites can contribute to sporulation. But then we also see that a lot of times they like to release their spores in the evening, so they might settle down quicker compared to when they're released during the day and being able to move between sites. And in general, a lot of these studies focus on bacteria just because they are smaller. They do tend to be aerosolized easier and can travel further than a lot of fungi in these communities. And one thing too, we don't look at it quite here, but is important are like the larger discrete events that happen. So storms, agricultural tilling, things like that can cause big bursts of these different microbes into the system as well. And a lot of that can be seasonal.
Okay. So I was thinking about that. So tilling can be a big deal here, right? I mean, it can really impact what's in the atmosphere. Is that right?
Yeah, for sure.
And do you see different, and I guess when I was thinking about it is this generally, so you would just be pushing up soil bugs, right? Sort of aerosolizing soil microbes. Is that kind of what you see when you see?
Yeah. And decomposers, right? Things that are on the plant material as well. And so that's where I think you get a lot of the fungi. You might get more bacteria that are in the soil, but then on the plant material, you'll probably have decomposers that are fungi in general.
Yeah. And I saw that, and I also saw that you sampled it day and night, and I think Carolyn kind of told me a little bit why, but I didn't know why. Like, why would you do that, right? Is it just simply because you think like fungal spores may be coming out at different times of the day? Is that what you're thinking, or?
Yeah, definitely. The biology is between ascomycetes, which are microfungi, and basidiomycetes, which are mushroom formers and rust and things like that. Mm-hmm. Their biologies are so different that we sort of expected that there might be a difference in when spores are released. So we know that with the basidiomycetes, the way that they release spores is they have to have moisture. They're forcibly discharged from the fruiting body, which is pretty amazing, but they have to have a droplet that helps them shoot off that spore. And so we kind of expected to see some variation in spore loads.
Okay.
Yeah, in different species.
And I think, I was actually talking to the folks in my lab about the paper, about the podcast today. And I was like, one of the kind of interesting things is, that I've never really thought about is how the atmosphere may be exchanging microbes, right? And stirring up the pot a little bit, right? And moving microbes around. But it seemed from your data that when you think about this, that there might be differences sort of between how fungi might be moved around in bacteria. Is that right? And what did you guys find, and sort of how did you study that?
Yeah, I think it was Well, it's hard to look at directly, but we can kind of see which factors they're kind of correlating with as far as what we looked at, mainly atmospheric factors, as well as some things like soil temperature. And the thing we found bacteria always generally being related to was strongly to wind patterns. So how strong the wind was blowing, what direction it was coming from. And that had a much larger effect on bacteria. We just kind of thought that a lot more bacteria are shared kind of across different sites, where the fungi seem to be much more specific to what was happening specifically in each of those sites. So they don't seem to be traveling as far. And if they do, they need to make it up higher to kind of make it between these sites.
And why is that?
That they need to make it up higher into the air.
Yeah.
Especially in the forest, because there's so much, let's say, like vegetation kind of density, they can easily stop in those areas and not be transferred. But it's quite wide open, at least in our grassland. So a lot more can kind of pass over that area.
And they are larger, right? They're heavier.
Yeah, they're much larger.
Especially the ascomycetes. So those are going to be a heavier spore. And so they need more wind, basically, and loft to get off the boundary layer and be able to lift up into the air. Yeah.
And are there certain species that when we should be thinking about that move around, or genus or general that move around a little bit better than others, or is it just all a size thing? Like how big you are kind of dictates how much you're moved around.
It's not. I mean, when we think about the fungi, we see a lot of Alternaria, Cladosporium, and we found those in our data sets, and those are our allergens. And so the spores actually of those two species aren't that small, but they are able to travel. So they do have hydrophobins on the outside of their spores that keep them really dry. And so they've been adapted to basically move through the air, and they're able to move up much higher. And we see in a lot of aerobiome studies that these two genera and genera like those, Alternaria and Cladosporium, are able to move around even across the oceans, which is pretty amazing, even though Basidiospore is much smaller than the ascospore of those species. Or those taxa.
Oh, cool. So there's been evolution, you think, to be able to disperse with in the air. Is that right?
Yeah, definitely. Yeah.
Oh, that's pretty cool. Yeah, I sort of thought, does this kind of mean that, the bacteria really the global generalists, right? And are the fungi more of a specialist, right? Is that the right way to think about it, or is that not the right way to think about it?
I feel like we see that a little bit. They do see in general bacteria that are kind of present across most sites we would consider generalists. We see a lot of proteobacteria and actinobacteria between these sites. That's pretty common, whether you're looking kind of in urban or these more natural sites. So for that, as far as the fungi, I'm not quite sure. Maybe Jane would be able to speak to that.
Yeah, I think there's some taxa that are very generalist that we find. And, fungi have evolved. Like one of the diseases that I work on is white pine blister rust, which is a Carnartium species. And we know it's able to get into the troposphere and move hundreds of miles from where the source is.
So how high is the troposphere, Jane? So
how high? 20. I don't know that actually how high it would be. I don't know exactly.
Okay, that's
fine. But much higher than the towers that we were at.
Much higher than the towers,
yeah,
right?
Much
that's
higher
what you're thinking, right? Okay, yeah, that's what I saw.
But basically, they're able to move, and so we know that maybe they're not as high as density as maybe the bacterial cells that we see, but definitely fungi have evolved to move through the air and to move long distances through the air, and we're able to capture them wherever we've looked thus far, right? The taxa that are there may differ a little bit, but there are groups that do much better moving through higher atmospheres, through higher altitudes than others. Yeah.
Okay, cool. The other word that I actually saw when I was reading the paper is vertical filtering. And I was like, kind of started looking this up, and I couldn't sort of. So, can you guys explain vertical filtering to me, what it means, and why we think about it?
Yeah, I guess I think in the context of this paper, since we have 3 heights, those are kind of what we're using as our kind of vertical layers, and thinking about, especially because we keep coming back to size. While it's not the only thing, it's a large thing that we've looked at. Basically how high they can be lifted, and the bigger they are, the quicker they're going to settle and really can't be lofted quite as high. for spores, of course, size, but then for bacteria, there's a lot of association with particulate matter, of course, coming up from soils or pieces of plants that are broken off. So kind of what's happening at each of these layers and just thinking about what could be preventing them from going as high. While maybe a single cell could reach higher, but will it actually survive? So that's kind of thinking about who's making it up there, what would be preventing them, and then could they actually survive when they actually get to that height?
Mm-hmm.
Yeah.
So
basically, you reduce your diversity, right, of taxa as the higher you go up, and that can be, like Carolyn mentioned, for many different reasons.
Yeah, it just seems interesting. So if you sort of get higher, you probably have a better chance of dispersing, right? I would imagine, right? You have a better chance. But are there actually negatives to getting higher also?
Mortality, right? So if you're not well adapted to it, then you die. Like it's a pretty dry environment. In the sunlight, the UV is very intense. So you have to be pretty evolutionarily adapted to be able to live in the atmosphere. I think it's a pretty harsh environment.
And in your study, one of the things that you saw was you did see what you think might be movement, right, from these two sites, from one to the other. Can you guys sort of elaborate on that a little bit about what you found? Yeah,
we mainly based off what is kind of shared between sites. Like we expect the basidiomycetes to be there. They're very commonly found in forest systems, where they're not commonly found as much in these grassland systems. And we found a lot of what was present in our forest system also present at our grassland. I mean, this could be between sharing sites, but we also are able to kind of model where the air had been and where it moved across these sites. So if it's not necessarily shared, there are air masses that could potentially be moving these same taxa between these sites. Mm-hmm. One thing we also thought was interesting, that we did see basidiomycetes at our grassland site that weren't present in our forest site. And we're able to model that at that specific time of day. It was like a PM July sample. And we could see that the air masses were moving from pretty much a different area. Okay. So we can get an idea of whether they're coming from that site or not, that there are shared taxa between these moving air masses across different sources.
And you guys focused on bacteria and fungi. Did you find viral sequences in this? Any viral sequences?
Yeah, we didn't look. Yeah.
No, we didn't. But that is definitely a big aspect of it as well. But we did not look specifically here. Yeah.
Yeah, because you might imagine that, even bacteriophage, right? I mean, you could imagine that they could be moving back and forth, and this might be a way to move around in the atmosphere.
Yeah, definitely. I mean, that's something you see all built in like urban environments, moving of viruses. So it's most likely happening out here as well.
Yeah. And you were saying that at least a lot of the microbes that are moving around are all of them, even the larger ones, on particles, or are they not? So
I don't think all of them are. I think maybe bacteria have a higher proportion, would be associated to some sort of particle. But I think for fungal spores, they're so well adapted to move through the air that I don't think they need a particle to do that. They're ornamented, they're melanized, they're thick-walled. They do have adaptations that allow them to move through the atmosphere a little bit better maybe than bacteria without a particle. Yeah.
And as you move up in heights, maybe not the heights, I guess, that you guys looked at, but maybe as the ones that are, maybe as you get higher up, I mean, are there issues with UV and other mutagens as you actually move up? Is that a concern, and where does that kind of start? Do we have an idea about where that might start?
I'm not sure where it starts. I mean, I think anytime they're exposed to full UV, that starts to happen. And we've done some studies in the lab looking at survivability of spores to different UV wavelengths. And basically it doesn't take long. I think we did 12 hours. Some spores are dead after 12 hours. Another spore, Carnartium rubicula, I think could survive 6 days. So really, we're not looking at long-term transport. I think a lot of this transport is pretty fast, like on a couple of days at most. And then they have to be able to land and find a host, right? Or find an environment that they're suitable in.
So if you're kind of riding the airwaves for too long, right, and moving around for too long, it's probably a bad thing. And so you probably need you get in the air, maybe disperse a little bit, and you have a day or so to get back onto something so you can germinate and grow.
Yeah, and I don't know for bacteria if they're on a particle, they may have longer shelf lives, right? They may be able to stay in the air a little bit longer, but definitely just the fungal spore in the air needs to find a home pretty quickly, I think.
Yeah. Yeah, so this looked like it also required some modeling aspects, right, for this paper. And because how much data did you generate? It looked like a lot.
It was a lot.
Yeah, it was quite a bit. We luckily had some atmospheric scientists we work with to be able to do the actual like air mass modeling. But as far as microbial data, we had a decent amount. Yeah.
Yeah. And so, when you kind of think about about this subject area and kind of zoom out, like, and I started thinking about like implications and what the average person might be thinking about, the thing I started thinking about, I mean, obviously, and I think Jane, you probably you work on this, is pathogen spread, right? Like plant pathogen spread, right? Is this like the primary way with which these pathogens are kind of moving around?
It depends on the pathogen, but if they are aerially dispersed, so for white pine blister rust, which is a rust pathogen, this is the primary mechanism that one of the spore stages is moved. And it's pretty amazing. We also have another campaign where we have set up basically aerial traps to try and figure out where white pine blister rust is moving in the west. And we're able to find white pine blister rust spores in sites that have no white pine blister rust. So we know, and these are pretty far away from where the nearest infected tree is, like hundreds of miles away.
Oh, wow.
So we know that this is definitely a good mechanism. There are some other fungi, this is a terrible mechanism, right? If they're produced in some sort of a mucus or they're wet spores and they need rain, insects and other, mechanisms to disperse, but definitely fungi, some of them are really good at this and moving through the air.
Yeah, and so the other thing I thought about when I thought about this was pathogen spread, but also as you're thinking about sort of temperature and climate change, right? I mean, as these things become as precipitation changes and temperatures change, right? I mean, do you think? I mean, is this significant? Does this impact the weather sufficiently that it could have a big effect later on?
It's a good question. I mean, we know that they interact with weather. I mean, especially precipitation, like clouds, as we mentioned. But as far as how that's going to change, I haven't looked into it as much. But you could assume that if more like discrete events are happening, certain storms or certain dust storms or like hurricanes, you get a lot more uptick. And especially they're usually more viable in these times compared to those that are just generally moving through the air. So if those can move and do, perform ecosystem services or do important things when they land somewhere, it definitely could have a big impact long term.
Yeah, I mean, I guess I was just thinking about areas that may be dry and start to get a lot more rain, right? And you might imagine that you have like, sort of blooms of fungi and microbes, right? That could then be, get into the atmosphere and impact, local weather and these things. Yeah.
Yeah, and I think a classic example are the allergens, right? So a dust storm event will push allergens across the oceans, and then people that receive the population that's dropped down, they get really sick from the allergens. And it's typically maybe strains of Alternaria that they haven't been exposed to, right? So their immune systems are a little bit more sensitive to those that have moved from large, sources from far away. So I think, yeah, we could have downstream differences or effects over time with differences in climate.
Yeah. And so I guess, this question is probably for Carolyn maybe first, is sort of, like when you started seeing the data come in, what was the first thing? Did something like immediately surprise you or make you think this was really interesting?
I think yes, because of course my background's more in soils. So of course you're used to seeing very big differences between sites, and getting more into the fungal diversity. That was what actually was the most striking, because I know less about it, but that's where we saw most of our changes and most of our very significant environmental impacts by site, where bacteria seem to be much more more shared. They're more, the ones that we're seeing are kind of ubiquitous. You kind of see them everywhere, and then we're seeing them across all these sites, where fungi seem to have just a much more local impact, which I thought was really interesting. And that's kind of what we leaned into.
Yeah. Well, how about you, Jane? Did anything surprise you at the beginning?
The coolest thing I think was the diurnal pattern between the spore loads, the basidios and the ascos, and then to find root decayers inside the grassland site was like, whoa, where are these coming? These basidiomycetes that are only found decaying wood, like woody material, which there is none in the grassland. And so to be able to do the back tracing and show that some of that maybe came from the forested site to the grassland, but also to show that the grassland site, the air before it hit the grassland site had actually gone up to southeastern Wyoming, where we have a forest there, and probably picked up some of those microbes, and then they were deposited into the grassland site. So it was kind of surprising to see just these taxa that you wouldn't expect in the air above a grassland. That was pretty neat.
Yeah, that's cool. So I guess we could start with Jane. I always like to ask towards the end, when you think about the field and, aerobiome research and over the next 5 to 10 years. So, I mean, what's the next hurdle? What's the next thing that we need to be doing?
We need to figure out if they're alive and what they're doing. Are they expressing genes while they're in the air? Do they go into some sort of resting state? I think a lot of the studies have really focused on who's there and how they're moving, less about functionality. So we do know that they are ice nucleating particles, right? So they're able to form basically moisture in the air, but we don't know anything else of what they're doing up there. Or is it just, there's this idea, is this an ecosystem where you have fungi there, or you have microbes that are always there? Or is it just a mechanism for transport? And if it is, then the microbes probably aren't expressing much, right? They're just shut down and then they move. And then what's their viability once they hit the spot that they're going to be in?
Yeah, and so what are the techniques? So what are the ways to do that? So how can you figure this out?
Yeah, so I'm part of a group called Broaden, which is an interdisciplinary group that's really looking at are proteins being produced? Is there expression? So trying to extract RNA from samples has been really tricky because you just don't have a lot of material. Right, the density of cells is so small. And so I think that's the next step, is figuring out, can we extract RNA? And then from that, what do we see? That's one of the things that I think needs to be focused on.
Yeah, I mean, I think we do a lot of RNA work from tough samples, and it can be tricky, right? I mean, it can be really hard to actually get enough out to do what you need to do, and especially when you have a lot of different organisms there, right? And trying to get an understanding more broadly of what's actually happening.
Yeah.
You said that folks were actually trying to figure out if they were actively expressing proteins. Is that right?
Yeah.
What's the method for that?
I'm not actually sure what the actual method is. I mean, you're doing proteomics, right? And
Okay.
They're trying to see what's available, but I'm not sure what the actual extraction process is after it comes off the filters. But that's the next step, is proteomics and metabolomics for this work. Yeah.
Yeah, I could see the metabolomics being interesting, right? If they're making certain metabolites, especially some that maybe have short half-lives, right? You may be able to sort of back calculate, right? And say, well, they must have made this, at these kinds of times. Well, what about you, Carolyn? What do you think about the next 5 to 10 years? Well, 5 to 10 years for you, what are you going to be doing?
Yeah, building on what Jane said, kind of how we have this group that's kind of working on it. One thing, too, that we're interested in is, okay, they're viable, then they get somewhere. Can they kind of establish themselves and do anything, especially ones that are landing in soils where it's very diverse and a very distinct community? If they get there, does anything happen? Especially with plant pathogens as well, landing on leaves, what's going to happen? So these are kind of the next steps. We can figure out who's there, are they active? And then when they get to these sites, can they actually have a large impact? Because it's thought they could affect diversity, but it's also kind of not known because we don't know if they can establish themselves in these new sites.
Yeah. But let's say that they're not viable. I mean, you guys would be bummed out probably if they're not viable, right? But let's say that they're not viable. Could it still be important even if they're not viable?
Yeah, I mean, from an allergy standpoint, you don't need the spores. The allergens are still allergens, whether or not they're alive. So there are some that are moving other sorts of chemicals around that might be important, right? Or other proteins or other things, yeah.
Yeah, I think they've seen something similar with bacteria. If there's toxins produced and they get to those sites, those aren't necessarily broken down, even if the cell may be dead. So if they're moving in large amounts, there could be still some definitely impacts.
And these physical impacts, right, like ice nucleation, these kind of things, those could still be occurring with a dead cell, I would assume. Is that right? Or do you need an active cell for those things?
I think you need an active cell.
Oh, do you? Okay.
Yeah.
Okay.
Yeah, I'd have to double check that, but yeah.
Yeah, so it seems interesting. I mean, I guess, for me, I just never really thought about, like the air around you. I mean, you do during allergy season here in Georgia, right? You feel miserable and, and all of those things, but you don't really think about how, as a microbiologist, how, the atmosphere may be moving organisms, vast distances, right, to new areas. And so, yeah, I thought the paper was really cool. And so I don't know if you guys have anything, sort of any parting remarks, you would like to say?
Well, I think more papers need to come out. So I hope I hope this paper inspires other people to start working on this, because the papers are far and few between, and there's so many questions we don't know yet. And so, yeah, hopefully people get inspired to do some more work.
Yeah, I agree. And it's similar to other fields where getting the terminology can be tough, like using aerobiome or bioaerosol. So it can make it hard to find papers across the field sometimes too. So always as more papers come along and it becomes kind of more all together, it'll definitely be easier to follow as well.
Yeah, it's kind of an interesting thing, I mean, when we sort of, a lot of grad students, as I travel around and talk to people, they're like, oh, there's so much competition. And I'm like, yeah, but it's nice to have a lot of people in the field, right? Like moving the field forward, because it really helps you. If you're stuck, right, a paper may come out that really inspires you, right? And go, that's what I need to do, right? And then your work actually moves forward. So I think competition it can be good, right? I think. And so the more papers that come out can be really good.
Yeah. And then they either confirm or reject what your data. I mean, for sure, other papers in the same field, I think, are super important.
Yeah.
Right? Yeah.
Yeah. Well, I mean, I think, the study kind of reminds us that the atmosphere is, I guess, not an empty space, right? We should be thinking about it. And it's an active conduit connecting sort of systems and moving things around. So, well, I want to thank both of you. I want to thank Jane and Carolyn, and thanks a lot for doing the podcast. And yeah, we're really excited to be here.
Yeah. Thank you so much for inviting us.
Yeah.
Yes. Thank you.
Yeah, and I just want to thank our listeners and tuning in to the Editors in Conversation, the mBio edition. And remember, mBio is a journal for the American Society for Microbiology. If you enjoyed the episode, please subscribe and share it with your colleagues. And until next time, I'm Marvin Whiteley, and we'll be talking microbiology again.